Hello. I thought I would just go through a few of these genetics problems. And excuse the mess, I had a lab here earlier today. So number 12, two individuals have these genotypes. How many different types of gametes can each produce, how many different types of gametes can each produce? Okay, yeah, they're each producing gametes. And what are the gametes? So I found this on YouTube, this formula. It says possible number of gametes equals 2 to the nth power, where n is the number of heterozygous gene pairs. So when you look at this, there's 1, 2, 3, 4 would be n. So 2 to the 4th power, that's 2 times 2 times 2 times 2 is 16. So 16 different types of gametes. And then it asks you to write down what they all are. What are they? And for me, this took a while. It took a while to figure out a type of system to do this. And by the way, every single one of these is misspelled. And it won't just change it because A, B, C, D apparently is not a word. And it doesn't ignore all the A, B, C, Ds. You have to just keep ignoring them. Those red lines drive me nuts. Okay, so I just did, you know that half of them are going to have a capital A and then half are going to have a lowercase a. And that didn't, Todd mentioned that. But then, so half of the big A's are going to have big B's and then half will be little B's. And then same thing with the little A's, half and half, so 4 of each. Now it gets tricky, but the C's, you go 2 big C, 2 little c, 2 big C, 2 little c on both sides. And then the big D is every other one, big D, little d, big D, little d, big D, little d, big D, little d. So these are our 16. I would worry it took me 2 tries at least to try to get these right. So you want to make sure that you can come up with those gametes. So 13, you set up a Punnett square using all the possible gametes for both individuals. How many offspring squares are in this Punnett square? And you already had this one. It was 16 times 16 is 16 across 16. And that's going to be 256. Given that the genes are all on separate pairs of homologous chromosomes, and that's really important, but all of them on this worksheet, they're all on separate pairs of homologous chromosomes. What method could you use to determine the probability of these individuals having an offspring that is big A, big B, little b, so het for those, little c, so that's homozygous recessive, and then heterozygous for big D, little d. And again, these are the parents from number 12. So you can go back and look, they were heterozygous for everything. Okay, there are 2 parents, heterozygous for A's, B's, C's, and D's. So the way to do this is to figure out a monohybrid cross for each gene, okay? So for the A's, here's a monohybrid cross for the A's. And they're both het, so big A, little a, big A, little a. They're all both hets that you started with because the parents were hets for everything. So say big B, little b, big B, little b, big C, little c, big C, little c. The C's look the same, so there's just a line drawn under the little C's. If you were handwriting it, I might draw a line right through it. And then big D, that should be a little d right there. And big D, little d. I can go back and fix that. But not right now because it's a whole picture. So the probability of having a het with the A's is going to be 0.5. The probability of having, let's see, a het with the B's is 0.5, big B, little b. The probability of having homozygous recessive for the C's is going to be 0.25. And then the probability heterozygous with the D's is going to be 0.5. So you just multiply all those probabilities together, 0.5 times 0.5 times 0.25 times 0.5, and you're going to get 0.03125 or 3.125%. I don't know if they want you to round it, but there you go. Now, what I like to do is check mine and Todd's answers. So Todd and I got the same answers for number 12, the same for 13, and so did you. And then for 14, we got the same answer and he did it the same way. Of course, it's a lot quicker to handwrite everything, but the 0.5 times 0.25 times 0.5, and he got the same answer. But I seem to run across some errors like that. So an organism that has the genotype or is heterozygous for A, B, and C is crossed with an organism that is homozygous dominant for the A's, and then it's hets for B's and C's. So here's hets for all three, and here is a homozygous dominant het. And now it wants to know what is the probability that any of the offspring will have the genotype big A, big A, big B, big B, big C, big C. Here's the hint. This is where it tells you how to do the problem. To get the answers, consider the 6 possible types of autosomal crosses. Determine the individual probability of getting a big A from a monohybrid cross. Do the same for big B, and big C, and then multiply the probabilities together. So that's what we did. We just looked at the A's here. Big A, and big A, little a. And then the big A goes down here, and then a big A across, a big A across. The little A's down here, and then we have big A, little a, big A, little a. So your probability is 0.5 of having a big A. Now for the 2 heterozygous B's, then your probability of a homozygous dominant B is 0.25. And then same thing with the C's, they're both heterozygous. So the homozygous dominant probability is 0.25. And you multiply those together and you get that same number from before, the 0.03125 or 3.125%. And then we can double check, that's number 15. Get Todd's 14 was the same, and his 15 is the same answer as mine. Now we go to 16. What is the probability that any of the offspring will have the genotype big A, little a, big B, and little c? Okay. Oh, I can't get those red lines off because that's all one picture. Well, so here's the original parents, and I might want to double check those original parents. Okay, het for all 3, and then one has hets for 2, except home for the big A, home for the big A, het. And then to double check it here, yep, you know, everything's het except for this one. And you can even use the same Punnett squares, except I wrote on mine and I just, highlighted them. I just unhighlighted them and re-highlighted the ones that we wanted. So we are wanting the probability of heterozygous A, which is 0.5, of heterozygous B, which is 0.5. Oh, you know what? Something's wrong here. Something is definitely wrong here because guess what? This should be a little a. So I'm going to pause my recording and fix it. If I can find my recording. I'll be right back. Okay, I'm not sure where I was, but I think I was on number 16. And I see that this is not an error if it's little a, big A, little a up here. times big A down here, okay? So perhaps I was on this one. And the probability of a hat will be 0.5. And then for the B's, we are going to hats, and then the probability of a hat would be 0.5. And then for the C's, we have heterozygous goats here, and you get one homozygous recessive. So that probability is 0.25. So 0.5 times 0.5 times 0.25 is 0.0625 or 6.25%. I better save that and save that. What I did fix was number 14. And then I looked at 16 and I was like, okay, that's not a problem. But there, the big D, little d. So for me, it's really hard, especially it's late, to keep track of some of these. So I have to look at these numbers over and over again. Also, I want to double check with Todd's just to see. He has 0.5, 0.5, 0.25, and then the 6.25% or 0.0625. So you want to make sure if they're asking probability or percent, if they have you handwrite it, you could write them both. So 17, consider the cross big A, big B, little b, big C, big D, excuse me, little d, big E, times big A, big B, little c, big D, big E, little e. What is the probability that any of the offspring will have het for A, homozygous dominant for B, het for C, het for D, homozygous dominant for E? Okay, so when we look at this, here is our cross. Double check it. Double check the next one. And we are looking for this. So, and then here's the big A, little a from here, and the big A from here. Big A's go down, big A's go across, little a's go down just on this row. So, and we're asking for this here, and that is 50% or 0.5. So the next one, we're looking at the B's. Here's big B, little b. Excuse me, here. Big B, little b, big B, big B. So your big B's go down here, little b go down here, and then big B's across. So we're looking for big B, and that's 50% of 0.5. Now your C's, we're looking for heterozygous C's, and we've got heterozygous and little c's. And half of them are going to be heterozygous. And we're looking for heterozygous. I was like, why are these underlined? But no, these are highlighted, and we're looking for hets. So here for the D's, we have little d's up here times big D's down here. So everyone gets a little d, everyone gets a big D. The probability of big D, little d is 1. No matter what, that's what they're going to get. That's 100%. And then for the E's, you've got big E, little e times big E, little e, 2 hets. And we're looking for 2 big E's. We have a 0.25 probability or 25% chance. So we're going to go 0.5 times 0.5 times 0.5 times 1 times 0.25. And we get that same number again, the 0.03125 or 3.125%. And that's 17. And you know what? Todd got the same thing. And looks like I would double check and make sure he has the same exact Punnett squares as me. but he got the same thing. So our next one is number 18. What is the probability that any of the offspring would have this genotype? And they're the same parents. So it'd be nice to double check big A, big B, little b, big C, little d, big E, little e. And I'm checking from Todd's paper. It's over on that other screen. Then we got big A, big B, little C, big D, and then big E, little e. So the probability of two big A's, we got the het, and we got the homozygous dominant, 50%. Probability of the big B's, we have the het here and the two, and the homozygous dominant. So here's the only hets here, and we have 50% big B. For the C's, we have a heterozygous one right up here and a homozygous recessive. And you know, nothing's highlighted because the probability of getting a big C is 0. And you know already from this point, if you're going to multiply in one of the numbers 0, it's going to be 0. There's no chance at all that you will ever get this genotype because you'll never get a big C out of that. Same thing with the big D. You have all you can get. Remember, we're big D, little d's. That's all you can get. So the probability of a big D is 0. And then the last, the probability of little e, you had a het here and a het there, so it's 0.25. Multiply it together and you get 0. And Todd has the same thing, which is why, and he has the same comment, none, genotypes big C, and big D are not possible. You know, it's so embarrassing in a class when you get someone who comes up with something that's not possible when you do the blood typing or something like that. Very embarrassing. You know, and like, well, these blood typing things are not really clinical, you know. So in the fruit flies, Drosophila melanogaster, then the most common eye color is red. A mutation or allele of the gene for eye color produces white eyes, and the gene is on the X chromosome. And just to show you basically how that's going to look like, here's a PowerPoint. Okay. Here's Drosophila melanogaster right here. And I hope I'm sharing the right screen. Stop share. And then I'll make sure to share it again. I could have been sharing the wrong screen the whole time. I don't know. It doesn't have the green line around it. Well, anyway, so here is the Drosophila melanogaster. And this right here should always be italicized because scientific names should always be italicized. So don't ever have it not italicized. But here they are. There's the most common. They have red eyes, and then they can have a mutation where they have white eyes. The gene for this is on the X chromosome. So when you see a female, they have 2 genes for it, so they can have 2 alleles. And the males, though, the Y chromosome is real short and it's missing a lot of genes. So this one is not on the Y chromosome at all. So whatever the male gets from the X chromosome, that's what it has. That's what he has. Okay, so here's, it looks like you have a normal male, and then it's mating with a white-eyed female. There's a female, and the W's look the same. Maybe if you're typing to a script or some other font or something, but there they are. Anyway, I just wanted to show you that. I prefer to do R's because with R's, at least you can tell the big from the little when you're handwriting. And this one's a red eye female and a white eye male. The other cross was not. It was a white eye female. So for question number 19, what is the probability that a heterozygous red-eyed female fruit fly with a white-eyed male will produce any white-eyed offspring? Okay, so right here is our heterozygous red-eyed female fruit fly. Okay, she's 2 X's, so she's female, and she's got one for red eye and one for white eye. And then she is mating with this male, so he's XY, and he only has one copy, and it's a little r, which means white. So here, the female here, X big R, X little r, and the male, X big R and Y. And then so you go across, here's the X big R, the X little r, here the X big R and the Y, and here the X little r from the male and the X little r from the female. So that is a colorblind female. And here the X little r from the female, and then the Y from the male. Okay, write the male sign next to it. But the probability of white-eyed offspring for this is 0.5 or 50%. Now, you want to pay attention because they might ask you what's the probability of a white-eyed male or a white-eyed female, or that one of the females will be white eyes. So number 20, what is the probability that the mating in part 1, so it's like the same thing, will produce any white-eyed females? And it produces 1 in 4, so 0.25 is the probability, or 25% of white-eyed females. What, and sometimes it'll say what is the probability that of the females one will be white eye, and then you have to go with other females. It would be 50% of the females are white eye. But what is the probability that this mating will produce any white-eyed males? Is 0.25 or 25% that this mating will produce any white-eyed males. Of the males, 50% though are white-eyed. So pay attention again to the wording. And look, it was the same mating. So I got to use the same Punnett square 3 times. Now this is tricky in a way. A heterozygous brown-eyed human female. Now my mother would be one of these. A heterozygous brown-eyed human female who is a carrier of color blindness. She's not a carrier of color blindness as far as I know. Marries a blue-eyed male. My dad's a blue-eyed male who is not colorblind. He's not colorblind as far as I know. Colorblind is a sex-linked trait. Colorblindness is, but eye color is not. So assume eye color is an autosomal trait and that brown is dominant over blue. Now eye color really is a lot more complicated than this because there's a lot of other different colors. But if you look at just brown and blue in the people I know, then it's pretty easy. So what is the probability that any of the offspring produced have the traits listed? And here's all the traits listed, okay? So I just looked here and I just thought, well, I'll figure these out myself. And here is the Punnett square for the color blindness. The female, X big C, X little c, she's carrying. And then the male is not colorblind, so he's X big CY. And then you can see out of 4, 1 in 4 is a normal female, 1 in 4 is a carrier female, 1 in 4 is a normal male, 1 in 4 is a colorblind male. And then you do this one separate. And that is the, because they said autosomal, so it's not going to be on the X and Y chromosome. So assume it's autosomal. So you get big B, little b. This would be like my mom. And I know that my mom, even though I can just see her phenotype is brown eyes, she must have a little b because her kids have blue eyes. Not all of them, just some of them. And then my dad has 2 little b's, so he has blue eyes. So when you look at this probability of brown eyes is, well, homozygous brown eyes 0, heterozygous brown eyes 50%, and homozygous blue eyes is 50%. So then what about the probability of brown eyes? 50%. The probability of blue eyes, 50%. The probability of color blindness, 25%. The probability of colorblind males. Now there is going to be a 25% chance of a colorblind male. Again, if they ask out of the males, how many will be colorblind? 50%. And that question's later on, but make sure you're clear. Brown-eyed colorblind females, 0%, because there are no colorblind females that will come from this cross. Okay, none at all, because the male is the father's not colorblind. So they're automatically going to get a non-colorblind from one of their alleles. Blue-eyed colorblind females, again, 0. If you were to say blue-eyed carrier, then you might multiply the probability of this one times the probability of this one, right? So 0.5 times 0.25 of a female carrier, but we're not doing that. They didn't ask it. So 0% chance. What is the probability that any of the males will be colorblind? So then you're just looking at the males. And when you look at the males, then half of them are colorblind. So 0.5 or 50%. Why do males show sex-linked traits more often than females? Because males are XY. So the gene or allele isn't going to be present on the Y chromosome in sex-linked traits. The Y chromosome is a little bitty chromosome. It's like missing a whole piece. So it's missing some genes, and those will be the sex-linked traits. And basically, you get usually in your autosomes, you'll get one allele from the mom and one allele from the dad. So if you have some type of recessive disorder like colorblindness, then you'll have the other allele to offset it. But since they only have one allele for it and it's on the X chromosome, then whatever they get on that X chromosome is what they get, nothing to offset it. Wonder what's wrong with this. Okay, we'll put that comma there. So I'm going to email this to you, and if you have any questions, then let me know. Good luck on your exam. Oh wait, one other thing. Hello. Say hello. Millie says hello. Millie says hello. Hi. I just woke her up. Okay. Go back to sleep. She didn't want to say hello, but she did it. Bye.